So for this third part, I really wanted to try to address some of the kind of biggest questions and the questions that are most frequently asked around, how do we decrease the costs? How do we scale up? And, when are we going to see mainstream commercialization? And in order to answer those questions, I think we first need to look at kind of where we are now. And just a brief timeline for those that are unfamiliar, this industry started really, I think, with Mark Post when he presented and created and presented the first cultivated meat burger that kind of launched the excitement of the industry and led to the birth of the first four companies in 2015. In 2017 was when we saw the first Series A fundraising rounds. And by the end of last year, there were six companies that reached Series A stage, and the ecosystem grew to, greater than 50 companies or so worldwide. At the beginning of this year's Memphis Meats raised, I think 186 million was the final total for their Series B raise round. And many companies have stated that they're sort of beginning to enter, either strike ground on the production of a pilot scale facility or actually have pilot scale runs that are coming online. So this is probably, somewhere in the 50 to 1,000 liter range, perhaps more for some companies, but somewhere around there, I think, is a good estimate in the terms of the volumes that the industry is currently at. And by all accounts, if you look at sort of the averages across the industry, it's, the price is still at least in the low hundreds of dollars per kilo. So if we look outward to 2025, I think a realistic outlook is, ideally having regulatory approval and products being sold in several different regions. And we'll talk about how I think a 10 to 100x cost reduction is definitely feasible in this short time frame. And then from there, it's, ideally by the end of the decade, we have really large scale operational commercial scale facilities that are operating on, 100 to 1,000 kiloliter volumes. But really right now, the industry is in this sort of prove it phase as these pilot scale facilities come online and really proving that, the technology that these companies are developing can scale and they can prove that they can lower the costs. So just to talk on costs, it's really focused on the cell culture medium. We understand that from surveying companies, around 60 to 90% of the marginal cost of the product comes from the cell culture media currently. And I just want to reiterate that, the question that we hear a lot is, is serum going to be used or is animal serum going to be used? I think the biggest point is that it's an economic non-starter. So the answer is no. And we've already seen several companies declare themselves serum free, either in the media or in other forms of journalistic outlets. And the problem is not that, creating serum-free medium is not, necessarily the hardest thing to do in the world, but the problem is, creating serum-free media still costs a lot of money. So we've been doing it for a while for a variety of different stem cells, but those medium formulations are still quite expensive. So the question is really, first stage, how do we get to serum-free? And then the second stage is how do we lower costs dramatically from there? And so there are really kind of four different areas of focus that I see in reducing the costs. And I'm going to focus on the first two of these just in the interest of time. But the first is really focused on the production of engineered proteins and growth factors and purified at food grade standards. So around 2 years ago, my colleague Liz Specht began this sort of medium cost analysis project using the medium formulation called Essential 8, which is a common stem cell media used for human pluripotent stem cells. And it's nice because it actually has just 8 ingredients. And so if you actually scale the costs per liter to 20,000 liters, you can see that, it goes from $376 per liter to over $7.5 million per liter. And if you look at this visually, you can see, it sticks out like a sore thumb that the driving costs here of the media are the growth factors that are produced recombinantly, and as well as other recombinant proteins like insulin and transferrin. And, these are basically essential components that cells need to survive, so you can't really eliminate them. But if you were able to really produce these at low cost, then some of the other ingredients become negligible. So the question is really, is this possible? So in Liz's analysis, which is again linked at the top and goes into more detail, ran through different scenarios in reducing the costs of the media. And if you skip ahead to this scenario D here, where you're producing all 4 of those recombinant proteins at $4 per gram, you can drop the price below $5 per liter. And that $4 price point was chosen because that's really what's realistic for the production of industrial enzymes that are used. So, again, fermentation is a mature technology. There's no, necessarily huge technological bottlenecks in producing proteins for lower costs at scale. It's just that we haven't really done it yet for cell culture media. From there, if you begin to source other of your bulk raw materials in food grade sourcing rather than pharma grade sourcing, you can drop costs below $1. And then finally optimizing some of the other remaining ingredients, you can get that to as low as 25 cents a liter in theory. So the price points below $1 per liter seem to be achievable without huge technological leaps. And a good example of this was an independent group at Northwestern University that earlier this year published a paper where, using that same medium formulation, they engineered the growth factors in the media and produced them all in non-animal hosts like E. coli. And doing so, again, lowers the cost because those feedstocks are relatively cheap. And they were able to reduce the cost of the media by 97%, down to about $10 per liter. And the remaining costs were driven by the insulin and transferrin that they were producing or purchasing from commercial entities. And so this is a really good example of how, just through engineering growth factors and producing them in-house, they were able to dramatically lower costs, just as a sort of experiment. And I think what's important to realize is that the proliferation phase of this industry, the growth factors that are needed to grow stem cells are generally well understood. And so there's probably around 12 to 20 total proteins that are going to be needed to service a lot of the proliferation phase of the industry. And so if we're able to produce those at very high scales, and then in turn at low costs, then you can dramatically lower the costs for the entire industry. So multiple B2B companies, as I mentioned, have already begun to work on media. Some of these companies are focused on whole formulations for specific species or cell types that cultivated meat companies are using. Some of these are making high value ingredients like growth factors, and some of them are even trying to find plant-based alternatives. But the infrastructure really already exists to scale protein and growth factor production. And so a lot of these companies like Richcore, for instance, that classically make recombinant proteins at pharma grade for the biopharma industry, are now starting to pay attention to the cultivated meat industry and producing things at food grade standards and lowering the costs. I imagine that other B2C companies may also opt to commercialize their cell culture media in the future, again, around that sort of IP protection idea that we discussed earlier. I think, a lot of the work that's gone on at companies has been around formula media optimization, and some of them may opt to actually commercialize that as the industry scales. So the second point is, from there, once you take care of the growth factor problem, the second cost driver is the other raw materials. So sourcing amino acids and sugars, vitamins, etc. So when we look at the raw material supply chain, going back to sort of across the whole alternative protein spectrum, this is where the sort of microorganisms, plants, algae, cyanobacteria, or other side stream biomass play a really critical role in reducing the costs. So you can use high molecular weight proteins from these microorganisms or other raw materials for use in plant-based meat replacement products. But you could also take hydrolysates of those to produce the amino acids that will be used to feed the cultivated meat industry. And then similarly, you can use the sugars from those to use in microbial fermentation, as well as the basis of scaffolding for cultivated meat production. So, things like alginate or cellulose are scaffolds that are abundant, or biomaterials that are abundant that can be used as scaffolding material in future products. So to illustrate this point, this is from a Reddit AMA by Blue Nalu, which is a company in San Diego, talking about optimizing their medium. And they say that, we can already produce the seafood that we're making without animal serum. So the question of scalability is actually a question of sourcing ingredients at low cost. So we've already seen that Blue Nalu and Mosa Meat have made early partnerships with companies like Nutraco, which operate, again, in this food and feed industry, to really optimize the raw material supply chains that can service them more efficiently. So switching gears to the question of scaling up, I'll walk through some brief infrastructure estimates and highlight some potential promising new technologies. But first, to look at this graph, we understand now that the current state of mammalian cell culture is generally in the field of therapeutics and biopharma, where you're producing really high value products in very low volumes. And we really need to work our way down this cost curve that's more in line with how food ingredients are produced at really high volumes and really low prices. So companies that are able to do this efficiently are poised not only to impact cultivated meat, but I think also translate those technologies perhaps outward of the industry into other industries like cell therapy or regenerative medicine that likewise need to scale. So just going to walk through some back of the envelope calculations to give you a sense of the requirements of the industry. And in general, most companies seem to be leaning towards this sort of traditional model that's used in biopharma of basically a seed train using stirred tank bioreactors to consecutively larger and larger tanks. And in this process, you could theoretically use the largest volume vessel at the end of your seed train line and harvest a percentage of those cells for the downstream process, either scaffolding, differentiation, et cetera, and then allow the residual cells in that bioreactor to double on the course of a day or two, such that you really have high efficiencies of cell production at the end stage in the high volume, and maximize, and then basically conduct several different harvests at different time points until you eventually have to clean and sterilize that line. So you can basically operate this on a semi-continuous manner, and by having semi-continuous processes operate in sort of staggered ways, you can kind of simulate a continuous process in a future factory. So if we're to do this and we're to simulate the volume requirements for a factory that's producing around 10,000 metric tons of meat per year, we're estimating that it will take around 100 of these 10,000 liter bioreactors to output that amount of biomass. Again, these are rough estimates and they're based on many variables, so don't read into it too much, but it's just a back of the envelope calculation. So if we return to the estimate by A.T. Kearney of, conquering 10% of the global meat market in a decade, that translates to 35 million metric tons of meat per year, which in turn translates to around 3,500 facilities that would need to be built, where each facility has 100 of these 10,000 liter bioreactor seed trains operating in parallel year round. So to give you a sense of what are those infrastructure needs look like for cultivated meat compared to what currently exists, you can see that the global production capacity based off of the survey last year was around 16.6 million liters, and the number of facilities was close to 1,700. So this suggests that we would need to create double the amount of facilities that currently exist globally in order to capture that 10% of the market. And similarly, a lot of these facilities that exist are significantly smaller in their volumetric capacity. So just as a very rough estimate, the needed volume capacity would be about 10 to 100x of what currently exists globally. So when we start, present these numbers to large groups of biomanufacturers and infrastructure people, this doesn't really scare them off. I think that the numbers there are something that they say, we have the materials for, we can build all of that. It's just a matter, I think, of the companies in this space being able to prove that they deserve that capital, that it's going to be required to actually build them. So I think that's a huge question that remains to be resolved and will be hopefully resolved over time. Now, given that these numbers are quite high, I think it's worth looking at other alternatives that would be able to lower the burden of infrastructure and costs on this industry. And so I just want to walk through a few technologies that I particularly like and some other people are pursuing that may resolve some of this infrastructure and cost issues. So one of the concepts that I like is about kind of going small to go big, where cells can be grown in different microenvironments that protects them from a lot of the problems associated with growth in stirred tank bioreactors, such as shear stress and nutrient and oxygen mass transfer. So for instance, in this example, you can see that cells are actually co-extruded along with a biomaterial like calcium alginate, where the cells actually are existing within these hollow core shell structures, and they're protected from shear stress by the actual hydrogel itself, and nutrients and oxygen are allowed to flow freely through that hydrogel. And cells actually really like this a lot because it mimics more of an in vivo state where, you know, they're packed together closely and they can grow in 3 dimensions and have their cell polarity and paracrine signaling, et cetera. And you can achieve very high densities of cells in these microtubes on the order of 500 million cells per milliliter. And to give you a sense of what that means, it's about, you know, 10x of what you might see in a stirred tank bioreactor. So by, you know, creating the larger number of cells in a lower volume, you can significantly perhaps lower the costs and also the size of the facilities. In addition, these cells in these microtubes are, you know, malleable. So, you know, you can theoretically have tubes that are filled with muscle or fat that are, you know, perhaps manufactured more similarly to how the textile industry is set up and eventually woven together in a sort of bottom-up additive manufacturing based construction method. And so this sort of eliminates the need for these sort of prefabricated scaffolds that would be, you know, somewhat difficult to seed with cells in the correct pattern that you'd want for muscle and fat that you'd find in an animal product. So I think that's one option that's interesting and deserving of more R&D attention in this space. Similarly, you know, another sort of established method of production is through hollow fiber membrane bioreactors, which operate on a fairly similar principle where you're having high density growth and low volume perfusion culture. The only difference is in this case, the cells are actually, you know, on the inside or outside of the lumen of these small hollow fibers, and media is then perfused through the fiber itself, and the sort of waste is flowed out the other end. And this is a sort of example from a paper published last year that illustrates the sort of volume requirements for production of 1 kilo of meat across different modes of bioreactors or types of bioreactors. And obviously with increasing cell density, it lowers the volumetric requirements to produce the same amount of biomass. So, you know, to be fair, these numbers for stirred tank reactors are not very generous. You can probably create a kilo of meat on significantly lower volumes, but still it illustrates the overall point of how important of a variable cell density is to, you know, achieving efficiencies here. And then finally, I think the concept of continuous processing is something that's talked a lot about in this industry. For instance, one company, Cellular Evolution, is developing a peptide coating that allows cells to adhere to and grow on, and eventually those enzymes that are secreted by the cells cleave sites within that peptide and allow them to kind of flow downstream in a continuous manner. And so the estimates for their footprint reduction in terms of the volume is around 7,000x, you know, compared to, I think, conventional stirred tank reactors. So all 3 of these sorts of methods, I think, rely on, you know, lowering the overall footprint in terms of the infrastructure and the volumetric needs in terms of the media, and producing a larger amount of cells on a lower footprint overall. So I wanted to end with, you know, we talked a bit about the challenges, but I wanted to circle back around, you know, the sort of opportunities that still exist in this industry, because there really are a lot. And so internally at GFI, we've been, you know, taking our sort of internal knowledge and engaging with a variety of different external stakeholders to map the sort of opportunities and white space, the R&D opportunities and white space opportunities for businesses to form across this value chain. And we'll be rolling these sorts of ideas out over the next several months or so, essentially drafted as sorts of one-pagers that explain the challenge and explain the concept of the idea, and really are looking for people to, you know, adopt these and drive them into new businesses or to undertake them as new R&D projects. So to give you a sense of what this is, you know, what it would look like is really mapping on different challenges across the value chain and white space ideas that still remain needed to be done for the industry. So there's plenty of things to do for each of these technology segments. You know, in many cases, we still don't have genomic sequences that are for the species of interest. And in turn, we don't have, you know, assays for cell identification. We don't have, mapping of genomic sites like safe harbor loci that may be used for gene editing and gene engineering of these cell lines. Around cell culture media, there's a lot of work that could be done around developing methods to recycle growth media, as well as leverage computationally assisted formulation discovery and optimization. For scaffolding, again, a lot of it goes back to really having fit for purpose needs of different assembly technologies like 3D printing and polymer spinning technologies that currently exist for other industries, but maybe need to be scaled accordingly or more fit for purpose for cultivated meat. Similarly, there's roles for computational scientists here in scaffolding to perform fluid dynamic models to understand the limitations of how fluids are perfused through porous scaffolds. And then the bioprocess design, again, kind of circles back again around this fit for purpose. What are the bioreactors going to look like if they do have scaffolds embedded within them? What is the harvesting process going to look like? How do we ensure that everything is reproducible, is sterile, et cetera? And then also there's a variety of different opportunities amongst the sort of technology aspects of the value chain. So for business and financing and business services, I think the idea of a co-manufacturing joint venture has been proposed as a way to increase the production capacity. So in a lot of other industries, once you get to the pilot scale, you can then kind of co-opt a co-manufacturer to produce the biomass that you need or the product that you need, but that doesn't exist in cultivated meat yet. There's a variety of different, safety, quality assurance, and regulatory issues that need to be settled to commercialize these products. A lot of research on, understanding the sorts of end stage product that's going to come off the line and what's its shelf life look like. The sort of microbiome, do cells need to have, or do products need to have a microbiome, or does that make them more susceptible to pathogenic infections when they're sitting in the fridge, even though they're sterile, for instance? And then there's a variety of other projects listed here. I won't read them all out. And again, we'll be rolling these out in more detail, but this is just to illustrate that, there's a ton of different ideas of how to accelerate this industry and to match the needs of the industry. And so there's no shortage of, I think, of good business opportunities that still exist. So the key takeaways for this section is really that, the field is nascent, but it's rapidly growing. I think there's a fairly clear path for significant cost reductions, but innovation in bioprocessing and scaffolding is really needed to reach the very large scales and decrease the burden of projected infrastructure requirements while meeting, the sort of product diversity that the industry is promising. And then finally, again, there's a ton of different opportunities that we've identified that will play a critical role in commercializing these future products. So thank you for listening, and I'll take any questions again.